Pardon me if this light causes me great problems, but it is exceptionally bright. So the slide deck, perfect. So for those of you that know me, I haven't spoken at HOPA in many years, so I'd like to thank the program committee and Jeremy and team for inviting me. It's a great honor to be here. I appreciate being able to speak at HOPA. It's a great opportunity to come and share some of the stuff that we're doing in our team, and also hopefully have a discussion afterwards about stuff that you're doing in your clinical practices. But for those of you that also haven't seen me in a while, since I have a beard, I'm just trying to be incognito, just in case you don't like what I'm saying. But also, if some of you are wondering why is somebody from South Dakota looking at my bio giving a talk on phase 1 early phase drugs, well, I did spend quite a few years in Kansas City, have a lot of good collaborations still around the country, and hopefully I can share with you some of those experiences. So my disclosures for the pharmaceutical employees in the room, this is not long enough. I plead with you. I'd ask for more research funding from all of you. I certainly want more phase 1 study funding, and would appreciate any opportunity to sit down and talk to you about some different combination strategies we'd like to employ. But I'm also receiving quite a bit of research funding for translational work. I oversee a translational lab as well as a sequencing lab, so I do have a fairly unique role in that I oversee both clinical practice, translational science, and also sequencing. And a shameless plug for our company, Vivify, it's out to set up an opportunity to interpret sequencing-guided treatment and help provide sequencing-guided treatment decision-making for clinicians at the bedside. So the objectives of this talk today, I'm going to basically set it up with, in the beginning of the talk, I'm going to talk about drugs that just got approved. Since I sent these slides in December, I kind of anticipated that the first few drugs would be approved by the time I spoke in March, and I was right. This last Monday, the approval of niraparib as anticipated, and then a couple weeks before ribociclib. So you'll see those as some of the first two drugs I discuss. But I'm also going to talk at the end about some of the new and upcoming interesting areas of science. I'm more interested in this talk in talking about the actual science and not necessarily individual compounds. I will describe some of the individual compounds in a few areas, but I'm actually going to talk more about drug classes and interesting areas that may be coming to the clinic in the next few years. So here again is a synopsis of what I'm going to cover, but I'm going to start with DNA damage and repair pathway drugs, and I'm going to talk to you about some of the cell cycle interesting pieces that you may have forgotten from pharmacy school and beyond. Briefly touch on PI3K/AKT/mTOR pathway drugs, specifically about a TORC1/2 inhibitor and PI3 kinase alpha combo. I'm going to very briefly have an immunology discussion. It's pretty much required that you include immunology in any talk and some of the state of the science, but I don't have enough time to talk today about all the different things going on in immuno-oncology. And then at the end, I'm going to talk about some new technologies and interesting agents, specifically about reducing resistance and also drug delivery for very hard to target targets, specifically proteins and other things that cause a lot of problems that we have a hard time delivering therapeutics to currently. So to start with, if all of you have read the Hanahan and Weinberg paper on the pathways, the hallmarks of cancer, I'm going to start with what I said to you in the PARP inhibitor domain, looking at DNA damage repair. So PARP inhibitors, when I submitted these slides and completed them, there was only one approved. When I was finishing the last revisions of my slides, there were 2 approved. So as of December 1st, there was only olaparib, and that was only approved for BRCA-mutant ovarian cancer. Multiple agents are still under study, as I'm going to show you in a minute, and in many diseases. Currently, we only have ovarian approvals, but it's going to come fast and furious in the next few years, maybe even the next few months, where you're going to see approvals or indications in breast cancer, prostate cancer, possibly lung cancer, glioblastoma, and other areas, and hopefully the ability to give them in clinic is not impaired by the access to these agents, because they're going to be challenging to give, and I'll talk about some of those issues in a minute. Several compounds, as I'm stating here next, rucaparib was approved in December, on December 19th, my birthday, and then niraparib was approved last Monday. And so combination studies with these agents are ongoing. Very rarely do you see a class of drugs that are relatively easy to combine like these. We combine them with a lot of things on clinical trials, and I'll describe to you here in a second some of them. So to start with, I'm not sure how many of you know the homologous recombination deficiency, or HRD score, and how much you use that in your clinical practice. But for prostate cancer, it's a big part of how they're deciding to give these treatments. And so for HRD pathway deficient patients that have a BRCA2 mutation perhaps, an ATM alteration, ATM, ataxia telangiectasia, a mutation, say that fast many times, FANCA, CHEK2, BRCA1, and several others that are listed there go into the HRD score. And there are several companies that produce an HRD assay. Foundation Medicine, using next generation sequencing technologies, have one, and I think that's the companion diagnostic for rucaparib. And then there's also Myriad and actually other companies creating them. And so there's going to be an ability to do HRD testing, HRD high or HRD low, to see which patients potentially could respond best. But those are most often used in single agent indications. And so when you're using these in the current setting, most of them are studied as single agents. But in the future, you will see multiple combinations with these agents. But in prostate cancer, it's definitely, I would refer to you to the Mateo paper from New England Journal from a few years ago. They're doing really fantastic work in the Stand Up to Cancer group, prostate group, and also many other centers around the country. And so some of the uses, particularly focusing on advanced ovarian cancer, you can see all of the agents listed here that are being studied at the moment. I have submitted applications for research funding and combination studies with all of these agents, for AstraZeneca, from Clovis, from Tesaro, from Medivation/Pfizer now, and also from AbbVie. And all these agents have a lot of opportunity for use in the clinic. They're all going after similar indications, and they all have fairly similar activities. The side effect profiles will also be similar, but you will see here in a second, especially for niraparib, how they decided to go after the maintenance indication. But some of the combinations that are currently under study that you will see in clinical practice in the near future are listed here. We're focused on combining it with immunotherapy and potentially other therapeutics. If you look at the right side of your screen, left for me, but combining it with chemotherapeutics, platinum agents specifically, especially for BRCA mutant patients, looking at CHEK1/2 inhibitors, CDK inhibitors may be very difficult to do, but CDK4/6 inhibitors may be very challenging based on the cumulative toxicities, but certainly the mechanism of action makes it interesting. PARP trapping, increasing that, especially for glioblastoma patients, and combining it with temozolomide, definitely appears to be an interesting opportunity. AbbVie has quite a bit of data accumulating with the veliparib and temozolomide combinations, but there's certainly no indication as of yet, and obviously that drug's not approved. And also combining it with taxanes for BRCA patients, combining it with VEGF inhibitors, EGFR inhibitors, all of the ones listed there. An ER-positive breast cancer with BRCA mutations potentially, and HER2-positive breast cancer patients, combining it with taxanes and trastuzumab plus anti-endocrine therapy. So here's the slides actually from last year's ESMO meeting that allowed niraparib, Tesaro's niraparib, to be approved for maintenance therapy in patients with recurrent breast cancer. I want to thank Tesaro for providing me these slides, but this was actually the slide deck that was presented last year in Copenhagen. And this was in platinum-sensitive, recurrent high-grade ovarian ovarian cancer. These patients had prior treatment with carbotaxol, but now they got carbotaxol again, and then based on their response to platinum treatment, they were randomized to either two arms, one with non-BRCA mutant and the other one with BRCA mutant. They were randomized 2-to-1 in each arm to niraparib maintenance or placebo, and then they were treated to progression of disease. And the primary endpoint was progression-free survival with a central blinded review. It was very interesting to look at the HRD positive patients versus the HRD negative, and also in the non-BRCA mutant patients. So again, BRCA mutant arm, non-BRCA mutant arm, but then that non-BRCA mutant arm was again separated into HRD high or HRD 0. If you look at the numbers, looking at the niraparib and the BRCA mutant side, 138 patients received the niraparib, 65 placebo. And if you look as of the data cutoff of 16.9 months, 47 of them were still getting therapy and only 4 that received placebo, so obviously significant. In the non-BRCA mutant side, the data was pretty similar. So looking at the BRCA mutant patients, the outcomes were pretty impressive. A PFS of 21 months for the niraparib patients and 5.5 months for placebo patients with a hazard ratio of 0.27. Very significant. Very significant. Obviously, you can see if the PFS has a median of 21 months that they have a long-term follow-up of at least 21 months. I think it was 22 months at this time, but the average was 16.9. For non-BRCA mutant, again, pretty impressive data, 9.3 months versus 3.9 with a hazard ratio of 0.45. And then for the non-BRCA but HRD positive patients, 12.9 to 3.8 with a hazard ratio of 0.38. So again, if you were not BRCA mutant but you did have a high HRD score, you had a significant likelihood of good response. And then for the patients that had HRD low, they also had a good chance of responding. I'm not including that data based on time, but I'll summarize it here at the end. Treatment-emergent grade 3/4 adverse events occurred fairly significantly. For those of you that use these agents routinely, you know that cytopenias are a significant challenge, especially if you give it more than cycled. If you give it single agent, again, most patients are tolerating it pretty well, but if you combine it with chemotherapeutics, it's pretty challenging to give unless you give growth factor support for many patients. Thrombocytopenia, anemia, neutropenia, all significant. Fatigue, pretty standard side effect that you see with patients with a significant hemoglobin drops, but only a small percent relatively to the overall percent of patients on the study. Hypertension was also fairly high, so for those of you that use these agents, make sure you're doing routine blood pressure monitoring in them. Many patients do have significant changes in their blood pressure. And also be aware, in the early studies, the FDA was mandating that bone marrows were done in anyone that looked suspicious for MDS. They were also doing standard MDS testing in many of the phase 1s, looking at how many patients do develop MDS. In this study, the percent of patients developing MDS was still small, 5 out of 367 patients that received niraparib versus the 2 out of 179 that received placebo. So not significantly more, but it's still a worry, at least to have in the back of your mind for patients receiving the PARP inhibitors. Dose adjustments due to treatment-related adverse events. Again, many of them were from the anemia, thrombocytopenia, neutropenia arm, but also several of them for fatigue. Grade 3/4 events that required a significant dose adjustment. Again, 2.4% for thrombocytopenia, so a majority of them were from anemia. So to conclude, from the niraparib data, niraparib significantly improves the PFS in patients with platinum-sensitive recurrent ovarian cancer, regardless of BRCA mutation or HRD status. And if you look at the data across the board, both for BRCA mutant, non-BRCA mutant, with HRD positive or HRD negative, I didn't include the HRD negative or HRD low patients because that was an exploratory endpoint, but you can see I'm summarizing the hazard ratio for you. It's 0.58. So niraparib seemed to work, and that's why you're going to see it in the indication, regardless of whether they were BRCA mutant or they had a high HRD score. The hematologic lab toxicities were most common, and they were very easily managed in these patients with dose adjustments, dropping the dose or holding the dose. And the majority of patients remained on treatment until disease progression. There was no detrimental effects in the quality of life. Actually, they did several very elegant studies for quality of life that will hopefully be in the manuscripts, but no significant changes were evident there. And I put in there on that last bullet a game changer. I'm not sure it's a game changer, but it's definitely going to change how we practice. Giving maintenance therapy for standard upfront ovarian cancer or recurrent ovarian cancer, I'm guessing that physicians will use this up front for the grade 3Cs and the 4s that get minimal or poorly debulked surgery with carbotaxol, I would not be surprised if they start using this in those patients. We'll see if it gets approved by insurance, but certainly for the recurrent patients, it makes sense to use a maintenance arm or maintenance treatment with niraparib as long as the toxicity is manageable for these patients. So first ARS question, please follow along and answer. I think it's quite funny if you have the slides open in front of you. I hate to say this, but when I submitted the slides, the lovely folks didn't remove my highlight. So in every one of these slides that you have in front of you, the answer is highlighted. So if anybody misses it, I'm coming after you. A few more seconds for people. Good. So for the 3, 2, and 5, for B, C, and D that did not have the handout in front of you, next time you'll know better. So look on the slides and you'll know. So the answer is A, fatigue. So the next part for the cell cycle that I'm going to focus on are the cyclin-dependent kinase inhibitors. So looking at the cell cycle, it's really important to remember the pharmacology of this and the basic science of how these drugs work. For me, in our translational group, we have several translational scientists that I work with that focus a majority of their efforts in understanding the cell cycle. This slide is actually from one of my translational scientist colleagues that works in our group. And Pradeep basically focuses on how the G1S checkpoint and how the G2M checkpoint really impact outcome. So taking a step back, for those of you that don't live in this world, one of the most commonly mutated areas of a cancer cell is the G1S checkpoint. The cancer cell wants to accumulate tons and tons of mistakes, and so it deregulates the G1S checkpoint. And so one of the targets, or the main target of what I just went over, PARP inhibitors try to take advantage of the fact that this is disrupted. The next drug class, the CDK4/6 inhibitors, are going to also target the G1 to S checkpoint. Later, when I talk about a WEE1 inhibitor, the AZD1775 from AstraZeneca, this is going to focus on the G2 to M checkpoint, and I'm going to revisit this again. But the G1 to S checkpoint is dysregulated or deregulated a lot, and because of that deregulation, the cancer cell is able to accumulate tons of mistakes. And because of those mistakes, it's able to do all the nasty things that cancer cells do, but the cancer cell has to then control the G2 to M checkpoint because it can't have so many mistakes that basically the cell is forced to die because there's too many mistakes. So it deregulates the G1 to S, accumulates tons of mistakes, and then allows the G2M checkpoint to be controlled, fixes enough of the mistakes, but not all of them, and then allow us to replicate, go through S phase as you would normally go through M phase as it normally would go through, and continue to propagate the cell. But if you can't control one of the checkpoints, the cancer cell is not able to become long-term viable. And so it's important to target one of these areas, and that's what these next therapeutic classes do. So CDK4/6 inhibitors. Currently, there's only one approved. As of 2 weeks ago, there's two. And as I was clairvoyant to say, ribociclib is likely to come in 2017, and abemaciclib is not very far behind. Most common area of study, as most all of you know, is breast cancer, but opportunities exist in multiple tumor types and in multiple combinations. We have phase 1s right now in lung cancer, combining it with immunotherapy and axitinib. And we'll definitely see other areas of focus coming where we're combining it with immunotherapy, PI3 kinase inhibitors, TORC1/2 inhibitors, everolimus, other things that are able to expand how these drugs are given, and I'm going to show you why in a minute. The chemical structures of these are important to know only from the standpoint that palbociclib and ribociclib are very similar. They're not identical, obviously. You can see the affinities for the different cyclin-dependent kinases, specifically 4 and 6. The nanomolar concentration of 4 and 6 is important to know. The difference is Lilly's abemaciclib, which is obviously not approved yet, is slightly different. The abemaciclib has, I'm hearing, I've never used it myself, so I can't say this, but I've seen the abstracts, I've seen the presentations, where the neutropenia, the hematologic side effects are fairly low. And so the potential to combine abemaciclib more easily with cytotoxics, or certainly drugs that cause anemia, thrombocytopenia, neutropenia may be far more opportune. The ribociclib, palbociclib, those drugs definitely cause a lot of hematologic toxicity, especially at standard doses. And so that's very much related to the affinity for the CDK4/6 pathway. But before I talk about ribociclib specifically, I'm going to talk about the potential combinations and why it's important. So if you look at the top of this slide, these are understanding or assuming that the PI3K pathway, mTOR pathway, plus RAS, RAF, MEK, ERK, all of them are aberrant in some way. And as a result of that aberrancy, cyclin E and cyclin D are upregulated, and p27 is downregulated. As a result of that, these CDKs can preferentially bind to cyclin E and then basically bypass the CDK4/6 mechanism. So if you have an aberrancy in the PI3K pathway, mTOR pathway, or the RAS, RAF, MEK, ERK pathway, it doesn't really matter what you do with CDK4/6. It just bypasses that, especially if the mutation or aberration is significant. And so I want you to understand that even if you're not sequencing at the moment, if you sequence patients using Foundation Medicine or any other test, certainly look and see if they have a PI3 kinase mutation, RAS, RAF, MEK, ERK pathway mutation. Most likely these CDK4/6 inhibitors are not going to be very successful in these patients. In an ER-positive breast cancer, a significant percentage of patients have a PI3 kinase pathway mutation. So it's potentially, certainly in a significant area of resistance or lack of efficacy, and you're going to see combination therapies as a result of this. The other part I want to mention to you is that ER is there, and that's downstream of the CDK4/6 pathway. So if you have an ER-positive disease and you're not blocking ER, it doesn't matter what you do to CDK4/6. You basically bypass the whole thing. It binds to cyclin D and goes through the S phase, and it doesn't matter. So please be aware, you have to block ER in combination with the CDK4/6 pathway when you have an ER-positive patient. I don't certainly know the data in other diseases besides breast at the moment, but I can assure you in breast cancer it's significantly important to do so. But in the lower panel there, if you block these, you are certainly reducing the potential resistance there and allowing for CDK4/6 blockade to be full and allowing for that cytotoxicity to happen as long as you're blocking ER and the potential to have a significant outcome that's positive for the patient. Now, again, not everyone's going to have problems with this. Even if you have a PI3 kinase mutation, not all of them are real, not all of them cause problems. I'm not saying everybody needs to go out and certainly sequence every patient, even though I personally think you should. In many cases, it's not necessary. However, be aware, if your patient's not responding to the CDK4/6 inhibitor, it certainly could be a result of these processes that I just described. So let's look at the ribociclib data and how it got approved. Many of you have probably seen this paper published last November in the New England Journal, looking at ribociclib combination with letrozole versus letrozole plus placebo, and for patients with hormone receptor positive advanced breast cancer. I want to point out to you this slide. I'm not going to include every bit of data. I refer you to the paper to look at everything in great detail if you have questions. But I think it's important to note in the first 2 months, you see the separation of the curves. And the separation happens very quickly, where you see a drop about 10 to 20% in the letrozole plus placebo group. So those patients declare themselves fairly quickly. And in the plenary session at ESMO, when this was presented, it was pointed out very clearly that this is going to be an area of focus moving forward for the advanced first-line setting for hormone receptor breast cancer, combining with the CDK4/6 inhibitor to maximize the early benefit then obviously that begs the question of how long do you keep it going based on the balance between toxicity and quality of life for the patient, and all of them are very important questions, and I certainly don't pretend to know the answer. But it's important to note again, you get the biggest bang for your buck early, then keeping the patient on obviously for 12 months potentially gives you a significant improvement in PFS and potentially overall survival long term. But looking at this, you can see the biggest separation at that first 2-month window, which is again maintained and only slightly grown over time. If you look at the subgroup analysis for the PFS outcome, you can see it was basically a slam dunk across the board for ribociclib plus letrozole. So the aromatase inhibitor plus ribociclib was significantly better across the board, except one place. Note for the ARS question coming up. For bone-only disease, it was not significant. So for patients that had bone-only disease, it did not improve outcome. There was a trend, but it certainly did not improve outcome. And for many metastatic or advanced patients with bone-only disease, it may not be the best option. It certainly is an option, but it may not be the best option. And the other point to note from this paper, for those of you that don't use the CDK4/6 inhibitors routinely, it's important to note that the amount of nausea, infections, fatigue, and diarrhea and alopecia that you see compared to the placebo group. We use these a lot. I would have never dreamed that this mechanism of action would cause this much nausea, but not a lot of grade 3 nausea, obviously, but a lot of grade 1, 2, and most of these patients do not get antiemetic therapy. So be aware they're at home, and lots of them, over 50% of patients are saying they're nauseated. So as a pharmacist, I think it's very important to note this. So talk to your patients that are on palbociclib, ribociclib. Talk to them. Are they having significant nausea? Based on the data that's out there, over 50% are. So it's important to be aware of. Also diarrhea, quite significant. Not grade 3 necessarily or grade 4 diarrhea, but significant amounts of diarrhea nonetheless, and much of it was early. Again, I leave the rest of it to your careful review, but the adverse events I think are important to note, especially for those of you that don't use these agents routinely. So the next ARS question, if you could please kindly take out your phone again. In which patient population in the Mona Lisa 2 trial, which is what I just showed you, was the progression-free survival statistically similar between letrozole and ribociclib plus letrozole? Again, if you don't get this one from the handout or me telling you, I will find you. All right, come on. Okay, C. Yes, that is correct. Bone-only disease. Again, the trend was toward improved outcome for the ribociclib plus letrozole group, but certainly it wasn't statistically significant. So I'm going to move on to the WEE1 inhibitor class. This is a compound in an area that is very exciting to me. I think this is certainly something that could be a big improvement for ovarian cancer and other tumor types, especially is an adjunct or a combination agent to other currently available therapies. I think the WEE1 PARP inhibitor combination is fairly attractive, as well as many others that I don't have time to talk about today, but certainly would be happy to if you have questions afterwards. But I want to make note of this slide, so please take a look at this. This is basically a pulled out section of the cell cycle, and impacting where this is in the cell cycle. This is in the G2M checkpoint, okay? So I talked about the G1S, and that is routinely aberrant and dysfunctional in most cancer cells. This then is far more on the G2 to M side, and as a result of this, cancer cells routinely hijack WEE1 through either a mutation in P53, which we all see. It's the most routinely mutated gene in cancer. Or you see ATM, ATR, which are members of the HRD pathway. So the homologous recombination deficiency pathway, these are altered routinely in many patients. So if you see a p53 mutation, you see this is going to be impacted. You see an ATR, ATM, CHEK2 mutation, this is going to be impacted. This basically then causes WEE1 to be the gatekeeper. This is a negative regulator. And if in the G10, G1S checkpoint is able to be completely altered, the cancer cell needs to control this as the gatekeeper. It needs to wait for enough mitotic delay for DNA repair to happen so that not too many mutations occur in the cell. They repair enough of them that the cancer cell can replicate itself and continue to live. If we alter this pathway, we basically force the cancer cell to replicate itself with all of those mutations inside. It's not able to function. It'll basically go through apoptosis. And so this is an important pathway. Obviously, there's much to be done to understand how it best works and how it's going to be combined with other chemotherapeutics. But certainly in p53 mutant patients or patients with HRD high, potentially in other mutations like CHEK1, CHEK2, which are also quite common, controlling WEE1 and being able to be really critically able to control the G2 to M checkpoint is going to be very important. And this is a very active pathway. There's not a lot of data to show you, but I thought it was important to discuss, at least be aware that the WEE1 pathway exists, and there's a lot of work going on in this. There is a little bit of data with AZD 1775 plus carboplatin in p53 mutant ovarian cancer. If you look at the plot, you can see in a lot of the patients. I know this is a very busy slide, and I refer you to it later. If you have questions, I'm happy to discuss it. I don't have a lot of time to go through this in great detail. But most of these patients that were done in this early phase trial were p53 mutant. They had serious ovarian cancer for the most part. A few of them were HRD deficient, but most of them that did have HRD deficiency, specifically the one that had the complete response there in blue, definitely benefited from the combination. There were a lot of toxicities from this. Again, this is early, but I think this is an exciting pathway that we need to exploit, and you'll see more research from this one coming in the future. Just a synopsis of the side effects from this combination. Most frequent all grade side effects, thrombocytopenia very high. Obviously a lot of that is from the carboplatin too, so we can't just attribute all of it to the AZD 1775, but certainly diarrhea and vomiting were quite high. But if you look at the grade 3 and 4 adverse effects, it was thrombocytopenia and neutropenia as you would anticipate, especially with the combination with carboplatin. And so I just want to make you aware of this pathway. I think it's exciting science. I applaud AstraZeneca for having this compound, and I'd love to play with it in my lab. So moving on to another aspect of the cell cycle that you may not have heard about. I don't know how many of you attended ASH, went to the ASH meeting this last year and heard about selinexor, but this is where I'm going next. And so the role of the tumor suppressor protein is another aspect of how the cell cycle can be manipulated. Tumor suppressor proteins function primarily in the nucleus to prevent the genesis of neoplastic clones, and this is also a way to regulate cell growth and the apoptosis pathway. Nuclear tumor suppressor proteins, the ones that are produced from the genes, are supposed to be balanced between what's in the nucleus, what stays there to have a suppressor function, and what's exported into the cytoplasm to do what it's going to do. So there's routinely a balance in our normal healthy cells where genes produce, ultimately through transcription and translation, the proteins that are done as action pieces to what the genes are expressing. And ultimately, this is done for a balanced purpose. Cancer cells export more tumor suppressor proteins from the nucleus to the cytoplasm and basically force them to not have a suppressor role. So you would expect to see p53, BRCA, and many other things as part of this process, and you're correct. So this is definitely another way to target TP53 mutations and other things that are very difficult to treat at the moment. The XPO1 transport protein is what they're specifically targeting. XPO1 inactivates these tumor suppressor proteins in cancer cells by stopping the routine export of these proteins from the nucleus into the cytoplasm. XPO1 is one of 8 known nuclear export proteins. It chaperones more than 200 proteins. It's the only export protein that transports these TSPs. There's no known redundancies in this. It's overexpressed in almost all cancers to date. And this correlates with very high likelihood of disease progression and a very poor prognosis, and potentially disease resistance in many patients. And so you will see this mechanism be tried to be exploited, at least in the future, and it's currently under study in many phase 1, phase 2s, and potentially some phase 3s in the future. Cancer cells love to inactivate TSPs, and the ones that are most likely to be inactivated are listed there, and they're all the bad actors, specifically P53, BRCA1, BRCA2, FOX1, FOX2, IKB, survivin, etc. So what's the role of the nuclear pore complex? Well, basically, this is the portal that XPO1 allows these proteins to be transported from their nucleus into the cytoplasm. You can read all the details in there, but the changes in this pore complex basically can dysregulate cell migration, intracellular signaling, DNA repair, cell division, etc. And the nuclear pore complex is unidirectional. It only allows the proteins to be exported, not imported. And so if you can't block it, basically these proteins are out in the cytoplasm forever. They don't come back in. And so you definitely have to have something that blocks the export of these proteins from the nucleus into the cytoplasm to stop this dysregulation from occurring. So by virtue of its non-redundant chaperone function, XPO1 is the target. And there are more than 14 distinct TSP or tumor suppressor protein pathways that exclusively use XPO1. And by inhibiting XPO1, it's certainly a viable strategy to restore the redundancy process that we need to have tumor suppressor proteins working in the nucleus to suppress the genetic function that's occurring. And so the selective inhibitors of nuclear transport is the drug class that I'm going to talk about here in a second. Cancer cells that overexpress XPO1, and we found ovarian cancer to be one of the highest, among others. Glioblastoma, pancreatic cancer, lung cancer are all equally good solid tumor targets. Then you have lymphomas, leukemias, myeloma. All of them are significant overexpressors as well. And if you look from the ASH data that I'm going to show you in a few minutes, the data looking at combination chemotherapy, standard induction therapy with the drug in this class, selinexor, was very significant and showed a significant benefit. Potentially in other diseases, myeloma, lymphoma, et cetera, are currently being studied. The selective inhibitor of nuclear transport compounds inhibit XPO1 from exporting these proteins, and it's certainly a potential great option to stop the migration of P53 and BRCA1, BRCA2, P27, et cetera. So KPT 330, selinexor is the name. There's another compound coming called verdinexor, I believe is the name of it. But selinexor is the one that's in clinical trials right now. It's the first oral SINE compound. It specifically reversibly binds to the nuclear export protein XPO1 to inhibit the export of 200 cargo proteins. It has a significantly well-established safety profile. I definitely know there are side effects. We haven't used it ourselves, so I can't speak to it myself, but I know that anemia, thrombocytopenia, et cetera, are significant challenges for some patients. But it's got a broad utility. It certainly has activity in solid and hematologic malignancies, and well over 1,000 patients have been treated as of December. And there are more than 40 clinical trials, and I'm sure that's growing in a broad range of diseases. So here are a list, and this is not an all-inclusive list, but here are a list of all of the proteins and the genes that correlate with those proteins. And it's, again, a lot of the bad actors. And if we can find a way to treat patients with P53 mutation with c-Myc, again, c-Myc is a transcription factor. How do you target c-Myc? Well, you certainly don't do it by stopping a direct target approach with a TKI like we have historically. And so we need another mechanism that potentially would be able to target c-Myc, and I'm going to talk again about another possibility later in the talk. But these are, again, areas that we need to work on, and potentially this is a compound or a class of compounds that could certainly be used, if not alone, in combination with other things, to reduce the burden of these patients that have these mutations. APC in colon cancer, again, is another opportunity to have an opportunity for treatment. And just a summary, the clinical trial results are summarized there. I have some of the abstract numbers listed in case you want to go back to the ASH website and pull some of the data. I didn't have time to summarize all of them. There were a lot of abstracts, and a lot of the data was quite good. But selinexor plus low-dose dexamethasone in patients with refractory myeloma, significant benefit. Selinexor and carfilzomib plus dex in relapsed refractory myeloma. Several of these were in part of the plenary session. Selinexor in combination with bortezomib dex in proteasome inhibitor refractory patients. And then combination with mitoxantrone and Ara-C in AML. Combination with Donna-Ara-C in newly diagnosed poor-risk AML, and also in elderly patients with decitabine. Certainly potential here in liquid tumors at least, and I think the solid tumor data is coming. So switching gears to the PI3K, AKT, mTOR pathway agents, there are over 30 agents under investigation in a variety of trials as of last November. I looked at them all up in clinicaltrials.gov. I don't have any chance here to list all of the agents. I'm not going to go through them all. I'm going to use a representative example that I actually have clinical experience with. But there are many agents, so I apologize to the companies that don't see their drug listed here. I apologize if you're looking for a specific PI3 kinase or AKT, mTOR pathway agent that you've used. I'm not going to talk about them. But what I'm going to talk about is the development of these inhibitors and how they've proven to be particularly difficult. It's not on any of your slides, but if you've done any of the trials with the PI3 kinase inhibitors, you know that they're quite toxic or have the potential to be. And a lot of the data that's been out there, if you look at the breast literature specifically, you will see that many of the outcomes were not what we were expecting. We were certainly expecting better outcomes. And a lot of that is because the patients can't tolerate the treatment. I think the dose and schedule of many of them are optimized for the PKs of the drugs individually, but based on the ability of the patient to tolerate them and the off-target effects of these drugs, they're significantly challenging for patients to take. And as a result of that, we obviously need to come up with more appropriate dosing schedules that maybe take advantage of the PK effects of these drugs, which potentially could be 1 or 2 doses a week, bigger doses less often, to maximize the benefit at the receptor and the downstream effects that happen from the TKI activities, or certainly combine them with agents that reduce the potential for resistance. There are lots of potential for resistance with PI3 kinase mutations, many downstream, upstream feedback loops that affect the PI3 kinase pathway, and certainly combining it with a dual agent like a PI3 kinase mTOR doublet seems very, very appealing. I know companies have come out with PI3K pan inhibitors. Those are quite toxic. Novartis has at least one. I think Genentech's had one. There's the PI3K mTOR doublet in a single agent. It's hard to regulate the dose of that, and so it's proven to be difficult to give. There's the PI3K alpha specific, there's the PI3K delta specific, there's the PI3 beta sparing. There's all kinds of different iterations of these, and they've all been proven very difficult to give. So I'm not going to go through all of them and show you just what I said, but again, they are difficult to give because of the downstream and feedback potential, and also the resistance mechanisms that occur. I'm including this diagram from, again, translational scientist in my lab to describe, some of the things that we're doing, and I'm not going to go through it. I use that as for your reference in case I want to specifically go back and look at what we're talking about. But the PI3 kinase pathway has many upstream and downstream impacts that do certainly impact how this drug class is given and how it responds in patients. So I'm going to specifically focus on the Takeda compounds TAK-228 and the TAK-117. And the TAK-228 is an orally available investigational early phase ATP competitive dual TORC1/2 inhibitor. If you look at the box on the right, it has basically no affinity for PI3K, but extremely high affinity for mTOR. And it has high affinity for both TORC1 and TORC2. In reference to the everolimus, temsirolimus, those are only partial inhibitors of TORC1, if you can see on the right side of your slide, or the left side, sorry, of your slide. You can see that they only affect TORC1. This inhibitor, TAK-228, affects both, and so it should reduce the potential for resistance here especially if the blockade is maximized. And so used in combination with an upstream PI3K alpha specific blocker, it seems to be a reasonable way to go. And that would be then potentially the TAC-117, which is the Takeda PI3K alpha specific inhibitor. If you look on the left side of the screen, the PI3K alpha is specifically inhibited. The PI3K beta, delta, and gamma are not, and the TORC1/2 is obviously not. So combining both the PI3K alpha specific with the TORC1/2 inhibitor potentially would be an option to block the pathway in total and to allow for this to potentially be a viable option, considering that we've had basically failures so far to date with the PI3 kinase alpha specific and pan drugs. So the design of this trial that was presented last year at the Munich AACR molecular targets meeting was the C32-001 study. This was a phase 1B, predominantly with Vanderbilt, and looking at how to give this combination. And I draw your attention to the PK/PD side, because I think it's important to note what I said previously is maximizing the dose by maximizing the PK/PD parameters, giving it less often, allowing for the side effects to be minimized, and maximizing the area under the curve so that the drug is able to do what it needs to do, but minimizing the toxicity. Again, this is the biggest challenge. So giving it less frequently at potentially a higher dose is going to be the strategy of developing these drugs in many instances moving forward. And looking at the early data, the adverse event profile is fairly class specific. For those of you that have given this before, you know that hyperglycemia is a significant side effect. I was a little bit startled to see that hyperglycemia was not a side effect here, which does give me a little pause because hyperglycemia is a result of the pathway being stopped. If you block the pathway, you're going to get hyperglycemia. I've used enough PI3 kinase inhibitors from many different companies to know that hyperglycemia is a major side effect. This study, I know that they gave patients very quick hooks with metformin, with sliding scale insulin, et cetera, so they didn't really describe a lot of hyperglycemia. Actually, it was quite low. But it's important to note that the drug-related AEs were significant, but most of them were grade 1 and 2. The targeting pathway reactivation, if you look at the PK/PD cutoff, you needed to get the drugs above the threshold for activity. And so looking at the dosing schema that was required, you needed to get at least 4 milligrams of the TAC-228 along with the duration per week of approximately 21 hours over the PD threshold. And so they did very elegant studies looking at how much drug they had to give and the time that they had to give them. And they basically settled on a treatment schedule of 4 milligrams of TAC-228 and 200 milligrams of TAC-117 every couple of days. And so they gave them basically 3 days a week. The outcomes of the phase 1 study that I'm talking about here, not overwhelming, certainly could be described as underwhelming potentially. They didn't have any sequencing guided data here, didn't show any specific PI3 kinase mutations. They took all comers. And so it's hard to comment on exactly the outcomes here, especially in a phase 1 study that was not really looking at that, only safety obviously, but there were no CRs or PRs observed in this study. There were a lot of stable disease, which is a positive certainly in this patient population, and many of the patients had stable disease longer than 3 months. So looking at the different formulations of this is again a challenge with this drug class and also with these compounds specifically, especially the TAC-228. They've had to reformulate it and basically looking at the different formulations of this compound. For those of you that are using it in your investigational pharmacy or in your practice on trial, definitely be aware that there are different formulations. If you look here specifically at hyperglycemia, only 5, 23, and 12% are noted. And that's why I'm showing you this side specifically. And the other side effects, especially the grade 3 or 4 drug-related side effects, very low, which is concerning to me that maybe we didn't get a high enough dose in. But certainly, again, this is a phase 1 study. There's many places to go with this. Just making you aware of it, that this is a combination that's coming. But looking at hyperglycemia as a side effect, make sure that you're aware that that probably is a class effect, and it should be there. So to summarize this, TAC-228 was the phase 2 dose from this phase 1 study. TAC-228 Monday, Wednesday, Friday, or 3 days a week, with TAC-117 of 3 days a week. The safety profile seemed to be reasonable. The PK/PD profiles were respectable, and certainly going forward, it looks like we have some opportunities in this drug doublet. We'll see what the data bear out. There are many studies, or several studies at least, going on. We have a phase 1 in our center combined with Taxol, and we're starting to hopefully open that trial in the next few weeks. So which of the following is an expected adverse effect of inhibiting the PI3 kinase pathway that may signify optimally hitting the desired target? I'm pretty good at giving away the answer routinely, so hopefully you know this one as well. Good. Yes, hyperglycemia would definitely be the answer. So very, very, very briefly, I'm going to discuss immune activating antibodies. Again, for those of you that have heard how many umpteen talks about immunotherapeutics over the last couple of years, read umpteen papers about immuno-oncology over the last several years, maybe a little bit sick of it. I'm certainly not sick of it, but I'm certainly feeling like we're hitting a little bit of a plateau in the outcomes, and I think that we need a slightly different approach at least. We know that PD-L1 is not a perfect biomarker. It is a biomarker that we use. I'm not going to badmouth it here, but I'm going to say that I don't personally think it's very good, but it is what we have. I think tumor mutational burden may be a better one. But certainly that requires a significant amount of work to be done. Looking at mutations per megabase certainly could be another one. Looking at a host of other things. But certainly knowing that PD-L1 expression is high does not guarantee your patient's going to respond. And we know in many patients that we see in clinical practice, they have a high PD-L1, they don't respond at all. And you're like, why? Well, there's many reasons why, and I don't have enough time to go through this, but I can certainly say that we need something to add to that to kickstart the immune system to do something. And the immune agonists are definitely part of that. So the T-cell agonists, specifically OX40, 41BB, GITR, IL-2 receptor, et cetera, all of them are going to be active components, and this is not an exhaustive list, so please don't take that for what it is. But there are many things going on in the T-cell agonist category that would complement the checkpoint blockage from a CTLA-4 antagonist, a checkpoint antagonist, PD-1, PD-L1 inhibitor, et cetera. And many companies are building this drug portfolio to take advantage of both the checkpoint blockade doublet, perhaps a single agent, plus the T-cell agonism that we need to definitely kickstart the immune system to be more active. And a lot of research is going on to see where this best fits. But knowing that we have, in many patients, hit a plateau where we can't get benefit in everyone. So some patients respond amazingly, even if they're PD-L 0. And so using a PD-L1 IHC test to say, yep, I'm going to give it to that person, or no, I'm not going to give it to that person, when you know in a lot of cases if you have a PD-L1 of 0, they're going to respond, and then the next patient you do, they have a PD-L1 that's off the chart and they don't respond at all, certainly that causes me great consternation. And so how do we then maximize the benefit, or what's the reason why? Do I have to count tumor infiltrating lymphocytes in the tumor? Do I have to do TIL counts in everybody? How do I maximize the benefit, or how do I better identify patients? And again, there's not enough time to go through all of this literature, but certainly be aware that the combinations of checkpoint inhibitors with T-cell agonists are there. They're being studied routinely, and the opportunity to find out where they best fit is certainly there. Combining it with chemotherapeutics, combining it with the next class of drugs that I'm going to talk about after this is going to be an opportunity. So bromodomain inhibitors and other drug delivery systems that reduce resistance to these drugs are also potential opportunities. And I just listed for you just the 4,1-BB combinations as of last November, and this was just a brief list. So there's 1, 2, 3, 4, 5, 6, 7, 8 different studies with just this in early phase. I could have done the same thing for OX40. I could have done the same thing for GITR. I could have done the same thing for many of them, and it would look very similar. So the number of studies being done here are exhaustive, and many of them are probably ongoing in your centers. So just be prepared for the agonism, checkpoint combinations, plus chemotherapeutics, plus radiation, plus many other things, plus PI3 kinase, plus many other pathways are coming. So in the last 20 minutes or so, last 15 minutes of this talk, I'm hopefully going to leave enough time for questions if you have them. But strategies to reduce drug resistance are a very important piece. It's a big passion of mine and a very big area of interest that we have on how do we reduce drug resistance overall. Combination therapy is one of the strategies that we're using, trying to shut down the escape paths of different treatments. And so when you use single agent treatment of metastatic disease, we all know that it's very rarely going to work. Now, we all have anecdotal cases where it does, and I'm not going to minimize those. We have in my practice. But the potential for third or fourth line treatment, single agent therapy, to be more than just giving them a little extra time is small. So clinical trials looking at combination strategies to shut down or slow down at minimum the escape pathways where patients are mutated in their cancer genome are extremely important. Epigenetic therapy is certainly a possibility here. I'm going to talk about one aspect of that, which is bromodomain inhibitors, and then I'm going to talk to you about a novel drug delivery system, induced protein degradation, or PROTACs, to finish up. So bromodomains. I don't know how many of you have used JQ1 or have seen JQ1 used. JQ1 was the first bromodomain inhibitor produced. It was produced in Jay Bradner's lab in Dana-Farber. Jay Bradner has done very well with JQ1 and has now moved on to, I think, leading basic science and translational research at Novartis. But because of his work with JQ1, which is the first bromodomain inhibitor, this is an extremely interesting and potentially very fruitful pathway that bears further study. But there are 42, I believe, bromodomain and proteins produced from 61 individual distinct bromodomains. The BET family proteins are the bromodomain and extracellular domain proteins. Specifically, there are 4. Bromodomain 2, BARD1 or BRD2, BRD3, BRD4, and BRDT. I'm going to specifically focus the next few minutes on BRD4, which is the most common. And what this does, there's 3 main mechanisms of what a bromodomain does. I didn't put it on your slide. I wanted to talk about it with you. It has a writing function, it has an erasing function, and it has a reading function. So think about bromodomains. They write, they erase, and they read. And this is all histone modification of epigenetic therapy. So the writers basically catalyze activity. They basically allow for the elongation and ultimately transcription and then translation of proteins to be made. These are recruited by cancer cells to basically control transcription. So if you control transcription, you control what proteins are made and ultimately what happens downstream of that. So for cancer cells, controlling this part of what a cancer cell's normal life cycle is very, very critical. The erasing function basically erases mistakes in the transcriptome, and then the reading function basically reads along, finds a mistake, and tries to fix it. BRD4 is a reader, and so what it does is it binds to acetyl groups that are closely linked to RNA polymerase II. And if they see an acetyl group that needs filling, they fill it and basically stop the transcription and ultimate gene expression, which then potentially leads to, if it's not stopped and it's mutant, increased resistance, increased proliferation, and decreases in apoptosis. Bromodomain inhibitors, on the other hand, are going to fill those acetyl groups and basically stop gene expression in its tracks, reduce resistance, hopefully drop proliferation, and increase apoptosis. And so bromodomain inhibitors are being really highly explored to reduce the resistance of treatment for everolimus or the PI3 pathways, mTOR 1-2 pathways, all of the different pathways that I described previously. They're being looked at to reduce the resistance of immunotherapeutics. They're being looked at to reduce the resistance of hormone therapy, especially androgen therapy. For those of you that are doing AR research, definitely a significant area of resistance in prostate cancer. So bromodomains are highly expressed in many tumor types. Some of the worst tumor types are overexpressed beyond belief. And so as a result of this is definitely an area for exploiting the undruggable currently. Here's not an exhaustive list, but c-MYC pops up again as a transcription factor and a regulatory gene expression marker, definitely something that we could target with acetylated histones. Other ones you can read essentially, but the androgen receptor I want to make potential note of there at the second from the bottom, really an important aspect of this. So transcriptional regulation of the AR target gene really could reduce resistance to androgen deprivation in prostate cancer and other diseases. And so this is definitely a significant area of research going on and a potential help for many, many men with androgen-resistant prostate cancer. Clinical trials of BET inhibitors are growing. There's not an exhaustive list here, but when I accessed this in early December, there are many, many ongoing trials with a variety of different drugs. So JQ1, again, was the first one, but there are many companies making a variety of drugs here. So I just want to make you aware of this extremely important drug class, which you will be seeing in clinical trials in the near future if you don't already have them at your site. And to finish up in the last few minutes, I want to talk about the proteolysis targeting chimeras, or PROTACs. I'm sure many of you have never heard of this, but what we need are better drug delivery systems. And what PROTACs are is a compound, a linker compound made by Arvinas Pharmaceuticals in, I believe, Massachusetts. It was started by a professor at Yale, Jim Cruz, and his team, but basically what they do is they find a target protein and they link it to an E3 ligase domain that basically tags that protein for ubiquitination and allows it to be basically dissolved and digested in the proteasome like it would be for a normal proteolysis process. And so this allows us to link E3 ligase to a target protein of your choice. You pick your target protein, and some of these target proteins could be scaffolding proteins that we don't have a target for. These could be targeting proteins of any kind. You use your imagination. So this is an opportunity that I think is going to be coming to the clinic in the near future. These are obviously big compounds. They're not easily made into oral dosage forms, but they're working on it. I have no stock in Arvinas, but I think I'd like to buy some. And as a result of that, they're probably going to do sub-Q or IV treatments for the near future, but I know they're working on an oral dosage form, perhaps a prodrug dosage form or something that they can allow to be orally absorbed. The timeline of this, it's pretty surprising. It's been around a long time, but fulvestrant was the first approved protein degrader, okay? So this is not a novel concept, but it's a novel delivery system for this concept. And so being able to target a specific protein, link it to an E3 ligase, tag it for ubiquitination and going through the proteasome and normal processes to get rid of it is an extremely provocative way to do things, and certainly something that I want you to be aware of. I'm not going to read this in there. I'm putting this in for your information, but I'm making you aware that this is, again, coming in the future. And to finish up, the mechanism of induced protein degradation technology that's here allows you to basically tag any protein of interest. So POI here means protein of interest. You tag whatever you want, use your imagination, come up with something, tag it with that E3 ligase, allow it to go through normal ubiquitination, and ultimately going through the proteasome and dissolving that protein like we would normally do. I saw this presented several times. I'm actually excited to go see another presentation this weekend at AACR in Washington, D.C., so I'm leaving from here tomorrow morning, flying home for 12 hours, changing clothes, washing my clothes, getting back on the plane, going to Washington, D.C. on Sunday. I know several other of you are doing something similar to that, but I'm excited to talk with the folks at Arvinas about potentially doing some research with these guys. But this is a definite interest to pharmacy because the pharmacology and the delivery system are our bailiwick, and certainly being aware that this is coming. Investigational pharmacists, beware. I'm very confident this will be challenging. And lastly, the proteins successfully targeted by degradation of this are lengthy. This is not a full list. But again, all the bad actors, AKT, AR, BCR-ABL, bromodomains, HER3, VHL1, VHL2, and the list goes on. PI3K, RAR, tau, many things that we don't have access to right now, we're not able to target because we just can't get a drug to it. Here's a potential for another one. So I talked to you today about several of those opportunities to target the untargetable, and that was really one of my intents today, to show you that we have potential for doing this in the future. So the last question of the day, which of the following is likely to be a potential target for protein degradation by a PROTAC? BRD4, ERBB3, BCR-ABL, or all of the above? Excellent. All of the above is correct. So in summary, and I thank you all for your attention today. I know it's late in the afternoon on Friday, the last full day of the conference, so it's exhausting to listen to me anyway, but especially at this time of day, I understand that. The next wave of PARP inhibitors and CDK4/6 inhibitors are coming, and they're already here. The PI3K TORC1/2 inhibitor, the PICTOR combination, plus chemotherapy is coming to the clinic. Obviously, it's investigational only, but it's certainly a viable option to better exploit the PI3 kinase AKT pathway. Immune combination therapy that I just briefly touched on, again, is a major emphasis for most companies and will continue to evolve, and I'm excited to see where this goes. And overcoming drug resistance via multiple mechanisms, specifically targeted protein degradation, is an exciting strategy for continued investigation. And I thank you very much for your attention. Thank you. Don't all of you rush to the microphone all at once, but I'm happy to take any questions. Okay, well, please come up afterwards if you have any. Thank you very much. Sure, I don't have one with me, but it should be in my box.